soil physical properties are represented in some way, and they do have an effect on how the organic matter behaves. So the upshot of this is that, well, I guess not the upshot. So this is all about sort of the heterogeneity of the material that's there, but also, as you could imagine, the turnover rate of that, how that material is being broken down and stabilized, is dependent on a whole lot of other factors, right? Because primarily there's both physical but primarily biological processes going on in soil. So they're subject to temperature and water and pH and oxygen and RH and all kinds of stuff. So that's where the kind of complexity comes into it. And I would say the overall thing, though, so it's both the complexity and all the factors that can affect these processes and the heterogeneity of the thing that you're trying to represent. And I think the biggest challenge really in modeling this soil organic matter is really to represent that heterogeneity. And there's kind of two different ways. I'll just to give you some, a little bit of background on it. And one is to say, okay, I've got heterogeneous material. Let me just lump it into boxes. Okay, I'll say I'm going to divide it into, old and young and ancient organic matter or something like that. Or I'm going to, subdivide it in some other way. And then there's kind of a lesser field of people that are trying to say, okay, let's accept that I've got sort of this infinite heterogeneity there and try to represent that heterogeneity rather as not as discrete compartments, but rather as a kind of a spectrum of quality. And so here's a couple of examples. This is called the Q model, is one that some Swedish scientists have published a lot on. And basically the idea of this, just to, and we're not going to be dealing with this later on, but the idea of this is you have an amount of organic matter, okay, in some arbitrary unit. You have an initial quality of organic matter. So think of plant residues. So in plant residues, you've got some sugars, you've got some proteins, you've got some lignin, you've got stuff like this. And those biomolecules vary according to their decomposability, okay? And it's more than just those. There's a whole lot. So you could say, okay, I have a quality spectrum of fresh organic matter that is defined some way, as going from high quality, which means very easily decomposed and metabolized by organisms, to lower quality. And so I have this spectrum. And the way I can model decomposition is both as the change in the mass as organic matter is decayed, but it's also decaying as a function of its quality. And as it's decomposed, the organic matter that is left behind is transformed in ways that makes it less decomposable sort of the more times it's going through a decomposition cycle. So anyway, the idea is that you can then model these changes as both, a loss in total mass, but also over time, a dispersion, a migration of this quality spectrum. And so that you can take this kind of mathematics then and turn that into a fairly elegant model of decomposition of heterogeneous substrates where the heterogeneity is described by this spectrum. Okay? Far more common, and maybe it's because these two guys, actually I would argue that that's not the reason, but probably one of the reasons, these two guys are both physicists who later got into biology. And so they wanted to make, more, theoretical, more elegant mathematical models. They say, well, it's very crude to take a heterogeneous material and divide it up into 5 boxes like the ecologists do. Let's envision this quality spectrum. But arguably, lots of people, and it's not that it's particularly hard to work with those models, but they've actually been less successful in a broader uptake, I would say. Anyway, the one that you're going to be dealing with is a much more, it's probably a little bit easier to conceptualize certainly the mathematics is that you say, okay, I've got plant litter, and some of that plant litter is things like cell walls of lignin and lignin in trained cellulose, and that kind of doesn't decompose all that fast. And then I've got, you know, proteins and sugars and all the kind of goodies on the inside of the cell, and I'm going to call that metabolic. And so when the plant litter dies, you know, I'll fractionate it into sort of two components, okay? And this one's going to decompose faster, this one's going to decompose slower. As those primary plant materials decompose, they are transformed into other bins of organic matter, okay? And, you know, so part of this quality spectrum is kind of like almost this cascade. As organic matter is decomposed, it tends to, you know, go into less available fractions, you know, as it goes through subsequent, you know, rounds of decomposition. And then these different pathways, which you can see, so decomposition is occurring both of the fresh plant material as well as the organic matter that's already in the soil from previous plant inputs. You know, everything is being decomposed. You're getting respiration and CO2 loss, but you're also getting residues that aren't fully mineralized to CO2 incorporated then into these other pools. And so this is simply a way of expressing that heterogeneity of soil organic matter. So it's a little bit more complex than a one-box model, but for the most part, if you look at the total soil carbon in aggregate, these individual components are very often modeled essentially as kind of first-order processes. Okay? So it's not too difficult. Okay, and then, you know, finally, this is kind of reiterating that a little bit, is to say that we're trying to model the soil organic matter. So it's both the decomposition, but it's also the stabilization of organic matter that isn't mineralized to CO2. Of course, decomposition depends on, you know, enzyme activities, you know, breaking down of extracellular materials, assimilation by the bacterial cells. Bacteria use that to produce their own biomass. They use it for energy, therefore CO2, but they produce secondary products, metabolites. The bacteria die, something else comes along, the fungi, etc. So there's all kinds of stuff going on there. And then you've got stabilization. So what is it that this organic matter that actually allows it to keep around in the soil for more than just, you know, a couple of years or something like that? You know, most organic materials, you put them in the lab, inoculated with bacteria, just about anything disappears fairly quickly. Okay, you can't have 1,000-year-old residues in those kind of situations. But so the other parts of trying to understand this is, you know, how do we represent the stabilization of materials? It has to do with how products are interacting with mineral surfaces or being included within micropores and aggregates in the systems and things like this. So that's kind of a little bit of a background of that representation of heterogeneity in the processes that are involved. And then each of those processes are subject to control of various environmental factors, okay? So yeah, I've got a rate of decomposition of a known type of organic matter under, you know, standard temperature, moisture, oxygen conditions. What if now I change the temperature? What if it gets drier? What if it gets wetter? What if the aeration changes? You know, microbes need nutrients, they need nitrogen, they need phosphorus, etc. So these are all factors that then get incorporated as kind of rate controlling parameters on those decay rates. So if we went back and said, okay, you know, I want to take one of these pools and I'm going to say, okay, I've got a decomposition process, so, you know, this is going off, some of it's going to CO2, some of it's going to this other, you know, this other pool, you know, pool A and pool B. Okay, I could describe this process as, you know, as some, you know, as part of, say, a differential equation, right? So this might be, you know, minus K times A. So I'd say that under idealized conditions, this is, you know, first order, it's like radioactive decay. So, you know, 0.02, you know, a fraction per year is the rate at which that decays, and if it were, you know, we would look something like this over time if that was the only thing I was looking at. Okay, but that, you know, that might be defined as a characteristic decomposition rate under, you know, ideal conditions, right? And that might be, you know, 30 degrees C, you know, some water potential, you know, 0.03, you know, megapascal, you know, pH equal to, you know, 6.5, et cetera, et cetera. Well, so the way to incorporate that mathematically is to say, yeah, there are, you know, rate controls that affect this, so I could end up saying that, you know, this rate here, this arrow that I'm representing here, is like this, but in fact, I have some function of temperature, some function of water, and maybe they're multiplicative, so if I solve that equation, then as I vary temperature, I vary water, I'm getting the response of the decomposition rate, okay? And one way that that's done in very many of these models is you say, okay, I need a temperature response function. If you define this constant here, this decay constant, as how fast it goes if conditions are perfect, then you might have a temperature response function that looks something like this, right? That would say, you know, so here's 30 degrees. If it's in the freezer, you know, decay rate is very slow. As I increase it, I get this kind of an Arrhenius, you know, Q10 increase, kind of exponential. I reach, you know, an optimum of 1 at, say, 30 degrees. And then, you know, and then if it gets even hotter, pretty soon the proteins and microbes start to denature, and, you know, and if you get to 60 degrees or something, then you don't have any more decomposition. You're, you know, you're cooking your litter instead of. So that's an example. And so you could do the same thing for temperature, water, pH, etc. So that's the way in which these kinds of things are put into these simulation models, which are simply systems of, you know, differential equations, often that are then solved numerically in, you know, in the model. And so here's, you know, this is a highly abstracted picture of this century model, and Descent is a kind of a version of this. It's a whole ecosystem model, okay? So plant growth, you have photosynthesis, organic matter, photosynthate, and nutrients are partitioned into different plant parts. You know, if it's a tree, it might look like this. A crop would have, you know, leaves, fruits, and roots, or something like that. You've got, you know, dead materials that come from the senescence of the plants. This stuff eventually decays and gets, you know, here was kind of a representation that I was showing before of different components of organic matter. You've also got a, you know, this doesn't show the water balance, but yeah, there's a portion of the model that is simulating the water balance, okay? So, you know, runoff and transpiration and all this kind of stuff. So it's all sort of an integrated ecosystem model, and that's what we're going to be using to do some of these you know, calculations. And this is showing a little bit more detail, just the soil organic matter part, and I guess a simple rendition of the plant production part. So you can see here, you know, if you start looking at